Welcome back. Hopefully you've taken a moment to either in your head or on paper fill in the different boxes we see here because it's critical that we understand how the process works properly before we can understand how it works in the context of disease. So first, either high or low blood sugar sends signals to the pancreas. And if we have high blood sugar, then the pancreas releases insulin. And insulin stimulates uptake from blood to tissue cells. And it also sends a message to the liver that glycogen and glucose need to be transformed into each other. So specifically with insulin, glucose needs to be transformed into glycogen. If we follow the blue arrow, if we have low blood sugar, then the alpha cells in the pancreas release glucagon, and this starts the glycogen to glucose pathway. This then releases sugar into the blood and raises blood sugar. So now we're going to look specifically at the light-colored boxes. So what do the arrows actually mean? So if you have high blood sugar, we're going to start with the box on the top right. Then it will promote insulin release by the pancreas, as we see going down the red arrow, and then insulin release will go both to tissue cells and to the liver, where it will stimulate glycogen formation from glucose. If you're working from the bottom with blue, low blood sugar, then the pancreas will promote the release of glucagon, and then glucagon will stimulate glycogen breakdown, so glycogen into glucose, and eventually raise blood sugar. So hopefully you at this point have a good understanding of how this system works properly. And so now we're going to start talking about what happens when the system breaks. Again, right now we're at homeostasis, and I'm wondering what you think you need to know about a disease. So in order to understand a disease, what list of things do you need to know about it? I created a list, and I think it's important that whenever you're learning about a disease to be able to answer all of these questions, at least in some detail. So first, we need to know how a system normally functions. So in this case, we're thinking about how the pancreas and liver and tissue cells all interact with insulin and glucagon to create a stable sugar metabolism mechanism. So keeping the appropriate amount of sugar in the blood and taking the sugar out of the blood and making it available to use for tissues. We're also thinking about physiology. So what these different structures are and how they work. Symptoms, so how someone might present with this disease. Diagnosis, so how do we determine someone has a disease? Treatment, so once we know somebody has a disease, what do we do about it? And I think it's really important to think about the larger context, right? So people are not just bodies that exist in space. They have families, they have cultures, they have a lot of different factors that go into them that aren't just about their cells. So I think it's important to think about everything going on around them that might have contributed to either their disease or to their health. So the two types of diabetes are called type 1 and type 2. I know it's really creative. And they have a lot of overlap. Their overlap is that they present in really similar ways. They have really similar symptoms, and that's why they're considered the same disease. However, they work by two different pathways. We're going to go into these in more depth, but the basic difference is that in type 1 diabetes, your body's unable to produce insulin. It either produces it in small quantities or not at all. In type 2 diabetes, your body can still produce insulin, but it doesn't use it efficiently. So insulin isn't able to do its job well because it's not able to bring glucose into cells. Type 1 diabetes is the minority of cases, only about 10%, whereas type 2 is the majority, so about 90% of cases. So later, when we talk about the global context of diabetes, we're thinking primarily about type 2 diabetes because it's 90% of cases. We're also thinking about type 2 diabetes because it's influenced by both environment and heredity. When I say environment, I mean the food that you eat, how much you exercise, other underlying health conditions like hypertension. Whereas in type 1 diabetes, the environment doesn't play a role. People are predisposed to type 1 diabetes almost entirely because of heredity. The final difference is that type 1 diabetes commonly presents in childhood, where type 2 diabetes most commonly presents over age 45. So children can get type 2 diabetes, but it's very rare for adults to get type 1 diabetes. So we're going to think more specifically about how the mechanisms of these two diseases work. First, type 1 diabetes is an autoimmune disease. So auto means self, and so it's part of the autoimmune system. And autoimmune means that the body attacks itself. So if we're going to zoom in, so here's our organ, the pancreas, and we zoom into these specific types of cells, the beta cells. In type 2 diabetes, beta cells are destroyed. And since beta cells are destroyed, and they are what signal insulin production, they start insulin production. When beta cells are destroyed, then insulin cannot be released. Since there's diminished insulin, if this is our fat or muscle cell on the inside and this is the outside, then glucose cannot come into the cell. So you don't have much insulin coming to these insulin receptors. The insulin receptors are signaling to the GLUT4 transporters to bring glucose in, but there's less glucose coming out of the blood and into tissues because of the lack of insulin. This happens because of the secretion of autoantibodies. And by autoantibodies, I mean antibodies that attack themselves. For a quick refresher on antigens and antibodies, previously we talked about antigens as any molecules capable of stimulating an immune response. So they have these epitopes on them that match with specific antibodies. Antibodies are produced by the immune system in order to attack specific antigens, and they recognize that specific epitope. In this case, diabetic autoantibodies or self-antibodies think that the beta cells in your body are antigens. So your immune system produces antibodies that specifically react to beta cells in the pancreas. So in doing so, they kill the beta cells and don't let insulin be released. So if we were to think about type 1 diabetes in the context of our initial diagram, we see that the place that the pathway breaks down is right here. The pancreas does not release any insulin at all or enough insulin in order for insulin to let sugar go into tissue cells or to stimulate glycogen formation. So this is the breakdown here because the pancreas is no longer making insulin. Type 2 diabetes is different. Type 2 diabetes is caused by insulin resistance and a damage in insulin receptors, which leads to a defect in signaling. So if we have type 2 diabetes, a person probably produces a normal amount of insulin. So a normal amount of insulin is released by the pancreas into the bloodstream. Then it comes to a muscle or fat cell, but there is a problem with the insulin receptors signaling to the GLUT4 pump here. In this problem, we don't actually get the glucose that should be coming into the cell. So there's less glucose than expected coming into the fat and muscle cell. This corresponds to diminished glucose intake, and so we see a lot of the same effects of type 1 diabetes, but it's a totally different mechanism. So the insulin receptors are not signaling to GLUT4, and in doing so, the fat and muscle cells are unable to take the glucose in the blood and put them in tissues. You would see how this would also correspond to high blood sugar because the glucose is not coming from the blood into the cells, it's staying in the blood. In our diagram, we see that the problem with type 2 diabetes is right here. So it's not that the body doesn't make enough insulin like in type 1 diabetes. It's that insulin can't come into tissue cells because of issues with signaling between the insulin receptors and GLUT4. So here is our problem. Insulin is not able to make tissue cells take in glucose. If you remember back to our lecture last week, we talked about the different things that insulin in cells and tissue will do. And there are 3 different things, right? There's glycogen synthesis. There's glycolysis, so it's taking glucose and turning it into ATP, which is the cellular currency that we've talked about, and finally fatty acid synthesis. So this means that people who have diabetes may lose weight really quickly, and we'll talk about that as a symptom in a little while. So, as we said just now, people can lose weight really quickly because, one, they're not able to use the sugar that they're intaking to turn into energy, and they have limited fat synthesis. This might seem counterintuitive given that type 2 diabetes is associated with obesity, but this still stands. People also tend to be really thirsty because, again, they're not getting the energy that they need from the food that they eat and the drinks that they drink. They may go to the toilet frequently because their levels of sugar are not being absorbed by the body, and that's really hard on the kidneys, and so the kidneys try to flush that out by quickly making people urinate, and that urine actually might smell sweet. It might smell like fruit. 3, people might be tired because, again, they're not getting energy from the food that they're eating. And if several of these are true, then a doctor might test for diabetes. What do we mean when we say test for diabetes? So there are 2 types of tests. If we're talking about type 1, then we can directly test for autoantibodies. So we can directly test those antibodies that are attacking the beta cells in the pancreas. For either type 1 or type 2, we can also do an oral glucose tolerance test. So an oral glucose tolerance test essentially means that you take someone who's fasting, they haven't eaten in a while, and you give them sugar. You then see how the body deals with that sugar. So for a normal person, you see that these are about the scores that they should have. So if they haven't been eating, then their blood sugar should be about 110 milligrams per deciliter or less 1 hour after they've eaten sugar. So they eat sugar here, 1 hour after eating sugar, then their blood sugar should be less than 160 milligrams per deciliter, and 2 hours later, it should fall down to about 140 milligrams per deciliter. To diagnose diabetes, people's fasting blood glucose levels should be over 126 milligrams per deciliter. And 2 hours after they've had sugar, then their blood sugar is still way up to 200 milligrams per deciliter. And so if you compare these two, you see that even given their higher baseline, right after they've had sugar, somebody's body is not able to regulate it if they have diabetes. We also have this column, which is impaired glucose tolerance or maybe prediabetes. As we've alluded to, type 2 diabetes has environmental factors. So someone can be at risk for diabetes because they may have a high body mass index. They may not eat healthily. They may have hypertension or another type of disease. And so they are somewhere in between what normal is considered and what diabetes is considered. So their doctor might prescribe that they change